Good morning to everybody. As previously said by Stefano, hepatitis delta virus is the smallest human virus to our knowledge that is a defective virus that needs of hepatitis B to end its life cycle. Regarding the genome of hepatitis delta virus is a negative sense single-stranded circular RNA that have a particularity of to be an enzyme. That means it is characterized by an autocatalytic activity because during the replication cycle, the polymerase generate a strand of RNA made by more than one viral genome. This autocatalytic activity allows the virus to separate the single genomes and to form the new viral particles. And specifically, the region that, I don't know, okay, that made this cut is the P1 region, while all the other regions are important to a proper fold and a proper functioning of the ribozyme. Moreover, the hepatitis delta genome encodes for only one antigen, the delta antigen, that is composed by different functional domains. And each domain have a proper role during the life cycle of the virus, from the translocation of the virus into the nucleus, the binding of the viral RNA, and then the coordination with the HBsAg for the release of the viral particles. Each functional domain is connected to the next by interdomains. But also the delta antigen is important for because play a role for the immunity that in fact contains the CTL domains that can stimulate the host immune response. Specifically the CD8 response that is important for the recognition of the infected hepatocyte and can lead their destruction. Globally, the hepatitis delta is worldwide, and we estimate a 5% of prevalence in HBsAg positive individuals. And due to its high genetic variability, have been characterized 8 genotypes, but the genotype 1 is the most represented in the world. And some of them are characterized by also by sub-genotypes. So, paucity of information is available regarding this genetic diversification. And so the aims of our study was to evaluate the genetic diversity of hepatitis delta, but by the point of view of delta antigen domains, CTL epitopes, and also of the ribozyme region. And we also evaluated the correlation of this genetic variability with biochemical markers. To achieve these aims, we involved 103 individuals followed in different Italian centers. And for each of them, we obtained a whole genome sequence by Illumina platform. And after the application of bioinformatic pipelines, we performed the phylogenetic and the mutational analysis. We consider only the mutation with an intrapatient prevalence higher than 5%, and also we define a nucleotide or amino acidic position conserved if we don't observe any mutation in the 99% of sequences. But move to the results, and these are the characteristics of the study population that is characterized by an overt HDV infection. In fact, the viral load, the delta viral load is of 6 log. The HBsAg levels are high, and also the ALT levels. Regarding the nationality of the individuals, they are mainly from Italy and East Europe, but we have also some African individuals. By analyzing the sequences, we observed that all the sequences belong to genotype 1, and we also observed all the subgenotypes of this genotype, and the most prevalent are the 1C and 1E. And this is in line with the nationality of our study population. In fact, the subgenotype 1C is most prevalent in individuals from Eastern Europe and the 1E from Italian. And also we have the representation of the two most subgenotypes that are mostly diffused in Africa, the 1A and the 1B. By analyzing the nucleotide sequence of the sequences of the ribozyme region, we observed a very high conservation, and it is in line with the most important role of this region for the viral replication. So any change in this in this region, can modify the proper folding and then in turn the proper activity of this ribozyme. By analyzing the aminoacidic mutational rate of the delta antigen, we observe a fluctuation in this rate in the different domains, interdomains, from a 27 to a 58%. The same fluctuation was observed for the CTL domains, but with a rate that never exceeded the 60%. By stratifying the population according to the two most abundant subgenotypes, we don't observe any differences in terms of mutational rate in the functional domains of the delta antigen between subgenotype 1C and 1E, and the same condition for the CTL domains. But if we stratify the individuals according to the viral load, we observe that individuals with a viremia higher than 5 log show the lower conservation in the CTL number 5, suggesting how a higher genetic variability can jeopardize, can hamper the activity of the immune system in recognize the virus, and in turn can enhance, can promote the viral fitness. Moreover, we characterized the mutational pathways of the subgenotype 1C and 1E, and we found the different mutations that are characteristics of these two subgenotypes. Notably, we observed that the co-presence of these three mutations, the 16V, 47E, and 202S in subgenotype 1C is associated with increased ALT levels, suggesting how an accumulation of this mutation can increase the hepatic damage. So in conclusion, from our study, we can say that the subgenotypes 1 of HDV are characterized by high genetic diversification, and the enrichment of mutation in delta antigen region can hamper the recognition of hepatitis delta from by immune response, and in turn can enhance the viral fitness. Moreover, the co-presence of specific mutation is associated with elevated ALT levels, suggesting how this mutation can potentially enhance the viral replication and in turn increase the liver inflammation. However, the role of this high genetic degree variability in affecting the proper activity of the diagnostic assay deserve further investigation. Thank you for your attention.